All right, in this video, I'd like to spend some time talking about this plasma membrane, this cell membrane. Now, this membrane is, of course, this boundary between the cell and the outside world, right? It is what separates the cell from other things. And it's also going to be this as a boundary. It is also how the cell is going to regulate what comes in and out of the cell. We'll spend a bit of time talking about that. And we'll also see that it is how the cell interacts with the outside world. So some of these little tails and structures that are hanging out of the cell end up being important in either sensing the environment or communicating out with that environment. So this cell membrane, this plasma membrane, is a phospholipid bilayer. And in the chemistry section, we talked about these phospholipids, right? They had a phosphate for the head and then these two fatty acid tails. We talked about how that phosphate group is actually hydrophilic, right? It loves or gets along with water. And so we see those hydrophilic phosphates either facing the cytoplasm, which is primarily water, or the extracellular fluid, which is primarily water. Now, who's tucked inside here are those hydrophobic tails, these fatty acids, right? Remember, fats and water, lipids and water don't mix. And so those fats are kind of hiding from the water that's surrounding them on both sides. And that is actually structurally what helps this be a really good barrier, right? So this phospholipid bilayer, or this cell membrane, is primarily a phospholipid bilayer, but they're also pointing out these big yellow blobs. That's cholesterol. Cholesterol helps give that cell membrane some fluidity, keeps it from being too stiff or too runny. And then again, as I mentioned in the last video, lots and lots of proteins on the surface of the cell here. Now, some of these proteins go all the way through the membrane, and they call those integral, or sometimes you'll see them called transmembrane proteins. And this indicates that the protein crosses all the way through the phospholipid bilayer. It is in contact with both the cytoplasm and the extracellular fluid. This is different than these peripheral proteins, which are only on one side or the other. And so those might be anchoring, maybe some of those act as anchors to the different cytoskeletal elements, right? Hold the membrane in place, that sort of thing. Now, integral versus peripheral is probably an important term, but more important, and definitely more interesting, is what all these proteins do when they're in the membrane. Now, I pulled this from another book, but I thought this was a really neat picture. So here we see a variety of transmembrane proteins, right? They're crossing all the way through. But look at the variety here. So this one, they're showing that some of these membrane proteins are receptors, right? Maybe a hormone receptor. Most hormones, when they try to talk to a cell, they actually dock on the surface and then a message is passed inside. Some of these proteins might be enzymes. When we get to the digestive system, we'll see that the last little piece of digestion actually takes place right on the cell membrane in the small intestine. We used an example in chemistry of lactase, and that is actually an enzyme embedded in the cell membrane. It does the final piece of digestion, breaking a disaccharide into monosaccharides so they can be absorbed. We will talk extensively about proteins that serve as ion channels, right? Allowing a variety of different substances very specifically, right? There is one protein that allows potassium through, a different protein that allows sodium through. Now, those channels can be open all the time for free passage, just depending on the concentration gradient of that ion, or it can be what's called a gated channel. So a gated channel is going to need something to actually trigger that opening. It could be something like a chemical, right? A neurotransmitter. We'll see this when neurons stimulate muscle. They actually pass chemicals to the gated channels on that muscle cell and tell them to open. So it could be a chemical. Sometimes it's actually a voltage that goes ahead and opens those channels, right? We have some proteins, particularly when we get into glycoproteins, and that is glyco indicates a sugar attached to that protein, right? So this glycoprotein is acting as a cell identity marker. This will be important when we get into the immune system and we see how things like white blood cells are able to tell self, right, our cells, do they have the right cell identity marker from foreign pathogens, say. This last one is a cell adhesion molecule, so they're pointing out that some of these proteins are actually helping to hold on to neighboring cells. This is actually really important for cells. Most cells need to basically be holding hands with their neighbor, right? Know that they're attached in their tissue in the proper place in order to keep functioning. The exception to this would be cancer cells. So cancer cells stop caring, right, if they're in the right place, if they're in the right tissue, and they're able to spread throughout the body or metastasize, which is one of the more dangerous aspects of that abnormal cell growth. Right? So a variety of different proteins on the surface of cells with a huge array of different functions. But your book points out this one that's actually, it's called a second messenger system. And I will say, don't spend time on this unless you're like super interested. When we get to the endocrine system, we can talk about it a little bit more. But all they're trying to show you here is that for some hormones, when they bind to that receptor, there's a whole cascade of events that actually transmit that message to the nucleus of the cell where it can be utilized. Okay, cell membranes are selectively permeable. Now in lab, you were using dialysis tubing, and that is semi-permeable, right? Some things were able to cross and others could not. A cell membrane is similar, but this idea of selective permeability, the cell membrane can actually adjust what it wants to let in or out, right? It can open and close channels. It's not a fixed substance. So this idea of selective permeability, we're saying that the cell membrane allows some things to cross and not others. Interestingly, to me, lipid-soluble molecules, here they're showing you, this looks like a cholesterol structure, so maybe a steroid hormone like testosterone or estrogen, is able to freely pass through the cell membrane, right? It is a lipid, and so it can actually dissolve through those fatty acid tails and pass through. The same is true, very importantly, lipid-soluble molecules like oxygen and carbon dioxide diffuse readily through the phospholipid bilayer, right? So those molecules do not need any kind of protein or any kind of help, okay? We also have a variety of water-soluble molecules that are able to move through different channels. We'll talk a lot more about channels. And then we also find that we have substances that are actually too big, right? We don't have channels that are large enough for them, and so they won't cross those cell membranes, or if they do, it's going to be an active process of engulfing them, right? So some kind of carrier mechanism. So the cell membrane, right, is selectively permeable. Let some things in, but is able to keep other things out. Now, let's spend a little time thinking about why or how these substances move through the membrane. Sorry, so figure 3-22 is a really nice overview, right? This one is a nice summary. Those of you who are more visual, if you're kind of zoning out as you're reading these paragraphs, focus in on this image, right? So we're going to talk about some of these processes which are passive. Passive means no energy, no ATP is used by the cell in order to achieve this outcome. Active processes require ATP. They require energy to complete the movement of this substance. So let's look at these passive processes. Okay, the first one that we should all become very familiar with is diffusion. So diffusion is the movement of substances from an area of high concentration to low concentration. So sometimes you might hear me say a substance is moving down its concentration gradient, right, in diffusion. So here we see, looks like all these little dots are CO2. We see a really high concentration, right? They're packed inside of the cell. And so sure enough, they're showing our arrows of diffusion that CO2 is going to diffuse out of the cell, right? A completely passive process. It's just trying to find a place with a little more elbow room, right, where it can spread out, okay? Now, diffusion works in all sorts of substances, and really what ends up affecting how quickly diffusion takes place is a variety of things, like how strong that gradient is, or how steep, maybe would be another way to say that gradient is. If you have really high concentration and really low concentration, things just go pouring out. If those concentrations are similar, it will move to that lower concentration, but that actually happens fairly slowly in that case, right? It can be things like the size of the molecule. Sometimes the molecule wants to move a direction based on diffusion, but maybe there's also an electrical charge that's pulling it one way or another. How lipid soluble they are, right? How freely they can move through the membrane, temperature, etc. Okay, so that is diffusion. A specific example of diffusion is osmosis. Right? So notice even in the definition here, osmosis is the diffusion of water. Okay? It is the diffusion of water across a semi-permeable membrane, right? And again, it always, because it's diffusion, is moving down its concentration gradient. So here's what tends to get students, we'll try to practice this in class is you're dealing with water and also a solute. Remember that solute was something dissolved into the water. When we talk about osmosis, we are only really interested in the movement of that water. So water wants to go from an area of high water concentration to low water concentration, right? So notice again, inside of the cell, lots of blue little dots, that's the water, less on the outside, and so the water's moving out. But notice what happens. The way you have a lower concentration of water on the outside is that there's lots of solutes there, right? And solutes, for most students, it's easiest just to think about something like table salt, sodium chloride, right? And so it is true that there is a high concentration of water inside of this cell, and so we see water moving to an area of low water concentration. But a lot of students, for some reason, really like this idea, and it's true, that water follows salt, right? Because notice what's out here is lots of solute, lots of salt, and water moves in that direction. If that helps you, by all means go for it. For me, for whatever reason, it's still much easier to think about high water concentration to lower water concentration. So just watch that as you're working through any of those problems. Okay, and again, this is only dealing with water, so a specialized form of diffusion. Okay, also a passive process, no energy required for water to move, and in fact, in your cells, as selectively permeable membranes, there is actually a very specific protein. It's called an aquaporin. All right, aqua for like water, agua, and then porin for pore. And so it's a channel and it's always open, and so water moves freely in and out of your cells. Okay, a couple more looks at osmosis here, just to make sure this is making sense for you, right? So what they're showing you here is like a tube, and it has this selectively permeable membrane at the bottom here. And what we're gonna see is that it's selectively permeable. It's allowing these little blue water molecules to cross, but the big pink solutes are unable to cross this membrane, right? And so, let's see, we've got two different concentrations, so I see a lot fewer solutes on this side than this side. Sure enough, there we go. Sure enough, what's going to happen is water goes from an area of high water concentration to lower water concentration, or water follows that salt, right? And so in this apparatus, you can actually see the water kind of drawing up, moving to the other side here. By the way, I have never seen this apparatus in real life. I'm sure it must exist. There's a reason that every textbook has a picture of this, but I have a feeling someone used it a long time ago to understand how water moved across membranes. Okay. This is interesting to me, right? We'll look at this, when we get to things like the kidney, this idea that you can actually oppose osmosis. Ooh, we get there in the cardiovascular system too. You can oppose that drive of osmosis by outside forces as well. So for example, if you have a bunch of sodium in your body, water will want to leave the cardiovascular system and maybe cause swelling, edema, right? So swollen ankles and things like that. But you could put on compression socks, right? You could apply an outside force and kind of keep that water from leaving the bloodstream and moving into the tissues, right? So there'll be a variety of different ways where we might see this come into play, but basically you have pressures. You can use pressure to oppose that movement as well. Okay. Now, in real cells, the way that osmosis becomes important is that, again, the cell membrane is this boundary separating the little inside world of the cell to the outside world. And what is outside of the cell, as far as solutes or water then, can make a big difference in the health of that cell. So look at what happens here. This is a cartoon of our red blood cell. This is an actual microscopic image, which is beautiful. And so what they're showing you here is we talk about these different solutions as far as tonicity, how much pressure they're kind of exerting on this membrane. If the solute concentrations are equal on the inside and the outside, we refer to that as an isotonic. We'll write that a little bigger. As an isotonic solution. So this is always a comparison from inside to out. And in this case, we're saying, oh, these are equal, right? And so what we see here is water moving in and out across the membrane at an equal pace. So that's also important to realize. Once there's like an equilibrium, it's not like nothing is moving, it's just that the ins and the outs are equal, right? And so that would be true for a red blood cell sitting in plasma, right? In your blood, that's kind of its environment. Okay. If I gave you an IV of distilled water, or if I took some blood cells out and put them in distilled water, and I watched what happened, that distilled water, that pure water, notice doesn't have any solutes, high concentration of water moves to low inside of the cell, and they managed to catch this picture of a swollen red blood cell. And what's remarkable about that picture is they caught it before the cell exploded, right? So a hypotonic solution refers to a very dilute. Hypo is lower. A dilute solution, this would cause water to rush in, causes the cells to swell, and potentially burst, right? That's not a good plan, which is why we don't give people IVs of distilled water. We actually give them an isotonic solution if they need fluids. Hypertonic solutions, right? Hyper, too much energy, right? Too much. So hypertonic is going to refer to a solution that has way more salt, well, not way more, it has more salt outside the cell than is inside of the cell, right? The converse of that then is that there's a higher concentration of water inside than there is out. And so water is going to want to move down its concentration gradient, and we actually see this cell shriveling, right? And the technical term in really small print right there is crenate, right? That means to shrivel. Over here, we'll actually see that cell explode, which is called lysis. Okay? Neither of these are fantastic situations, right? And so cells would love to be, whenever possible, in an isotonic solution. Okay. The last thing that fits under our passive membrane transport is what's called facilitated diffusion. Now, this one is the one that trips students up. Here is your key, right? It is diffusion. That makes it passive, right? No energy required. What throws students off is this idea of facilitation. So what this refers to is this is going to be things like ions that need a protein in order to enter the cell, right? They're not just flowing freely through the, say, the phospholipid bilayer or through just a constantly open channel, okay? But in this case, nutrients, the example they're giving is glucose here. It's moving across the membrane. It is always still moving down its concentration gradient. And what throws people off is, like, it looks like this protein is doing work, but it's actually not. When glucose binds on an active site, right, on this receptor, without any energy, it's changing the chemical bonds in that protein and causing it to kind of tip open the other direction and dropping that inside. Right? And again, it's always about the concentration gradient from high to low. It's diffusion, so it's passive. Okay, ask me in class if you have more questions on that one. All right, so those are our passive processes. Let's hit active and then we'll take a quick break here. So our active processes, again, require ATP. The one that most people are familiar with, this is the classic example, it's called the sodium-potassium pump, right? And what happens here is the cell is essentially always bailing itself out. So it's kicking 3 sodiums from inside of the cell to out and bringing 2 potassiums from outside to in. But this is against the concentration gradient. So notice there's lots of orange dots, lots of potassium inside of the cell. That is actually the cation that the cell prefers to have in the cytoplasm. But it's using energy to pump against the concentration gradient and bring more of those orange potassiums in. Same thing is true over here. We see a lot more sodium outside, but we are still pumping sodium to the outside of the cell. Now, there are other examples of this as well, and one of the things that we'll see is you really need enough of these proteins in order to move whatever that substance is that you would like to move. They can get maxed out, and then we also need enough energy, enough cellular energy in the form of ATP. But this is the classic example. This is the one you'll see most, okay? Active transport. And isn't that fun? It just tells you in the name that it's active. Okay? Otherwise, we're looking at using energy to do more of what we would call like a bulk movement. So a couple examples here of endocytosis. Endocytosis is basically the cell bringing things in. It's the packaging of extracellular materials into a vesicle for transport into the cell. So let's look at what that means. Here's our phospholipid bilayer kind of dipping in, and it'll eventually pinch itself off and bring that in. Now, receptor-mediated endocytosis don't worry about it too much, but it's super cool that you actually have these little receptors studded, right? Little proteins sticking in the cell membrane, and they're grabbing onto specific molecules as they come by. And then once all those receptors get filled, the cell membrane pinches off, and we bring that into the cell. Pinocytosis is an example of endocytosis that we would say is cell drinking. So the cell kind of, again, forming that little vesicle to bring a liquid in, versus phagocytosis, which you probably have heard of and we see a lot, where we're actually, the cell is engulfing something solid. This would be an example here of a white blood cell, like a macrophage, grabbing maybe a bacteria and grinding it up inside of the cell. All three of these are endocytosis, bringing things into the cell, and they all require ATP. Exocytosis is just the reverse. It's taking that vesicle, actually fusing it to the cell membrane, and dumping that substance to the outside, right? So this might be a way that you get rid of cellular debris, different wastes, but again, it requires energy. Okay, let's take a quick break.